Very good evening to all of you. First of all, I would like to thank the organizer for inviting me and giving me an opportunity to present my work since last 15 years, what we are doing at our laboratory. I'll be talking today on molecular approaches, navigating the future of health science. At our laboratory, this is what we do. We identify new technologies, new molecular methods, new biomarkers, which will help the clinicians to take a perfect decision to ensure that they are giving us appropriate treatment line and also keeping the patient's safety into concern. So I would like to start with my talk with a very known historical witness that we have done, that we have seen, that definition of power. A century back, the definition of power was muscle and physical strength, but it changed to wealth in some time. Wealth is equals to power. But now, in current trend, my friends, power is your knowledge. The more the knowledge you have, the more the power. Why I'm emphasizing on knowledge is I'll end with my talk with also knowledge. And to support my statement, this is a very good must-read book from Melvin, and it's a very beautifully explained book where the knowledge and the impact of knowledge on the current power. So when I'm talking about knowledge, it's a very vast definition. It's a very vast definition from inwards and outwards. So as inwards as we go to up to one cell of the body, the protein interaction, which I can see in the right-hand side of the picture, and when we go outwards to the globe and the internet connectivity, both look so complex, so variable. So that also gives us a space that more knowledge has to be acquired, more understanding is required, and more we understand, it can be applied for human and for better mankind. So I'll be restricting myself to talk about more on healthcare, and whenever we say healthcare, we say something is not right also. So whenever we are feeling low, we are feeling not healthy, there are a few questions which grumble our head saying what it is, how, what should I do, what treatment line I should go for, or what else I should be thinking of. And we go to the clinician. When we go to the clinician, it's the same question the clinician answers to us. Okay, Mr. or Miss, you go for this test, you go for this treatment line, this is the better approach, based on their experience and their expertise and their specialization that they have worked in since last many years. So here, I will be more talking today on molecular approach on these questions, and specifically to reproductive health. Why? Because I've been working on reproductive health since last 20 years. I've been also working associated with cardiovascular disease, with neurological disease and oncology, cancer disease. But to give respect to the time, I'll restrict all my examples, all three examples that I've taken today, to reproductive health. So the first question, what test should be done? What kind of test should be done to understand the problem? So I would like to give an example of one of the experiments that we did. Whenever a female goes to an IVF center or an infertility clinic, she's subjected to ovarian stimulation. When I say ovarian stimulation, the female is made to make more eggs in that particular cycle as compared to the natural cycle, which is just one egg at a time. And that is done by a commercially available hormone called follicle stimulating hormone and luteinizing hormone. Now these hormones, they bind to the receptors on the cell surface and bring about transduction of messages inside the nucleus, produce RNA protein, and that's how the cell responds and the ovarian stimulation takes place. We understood this, that this receptor, as it looks like very large in number in the sequence, and it has multiple polymorphism. When I say polymorphism, multiple changes. So this changes makes that particular receptor in changing their capacity or having their affinity to bind with the hormone or not bind with the hormone. How does it transcribe to us? How does it help us? We sequenced it, all these receptors and the associated genes, understood the variation and grouped it. We did around 200 odd patients and tried to understand whether the response and the sequence, can they be connected? Can these two different information intercalated? And when we did this, this was the response, this was the outcome. I'll spare you with all the details, but what you need to understand is this. So based on the sequence, you can classify or categorize females as good responder, moderate responder, and poor responder. So what does that mean? When she's a good responder, give her a little dose, moderate responder give at a higher dose, and a poor responder give much more higher dose. So this is an example where a female, a donor, was first time given 400 units of FSH. She gave good number of eggs and embryo. Then we did the sequence, and we found she's a very good responder. Why should we give this much of hormone? We translated to 200 unit in the next cycle, and you just imagine she produced the same quality and the same number of oocytes. So we could save this female from hyperstimulation, from subjecting to hyperstimulation of ovary, and that can help us to help the dose of the FSH and LH hormone to go down. And this example is opposite to that, where the female was given her 200 unit for some reason, but she did not well, the cycle failed, entire cycle has to be started, and then when we did the sequence, we found that she is a moderate responder, we should give a higher dose. So why I'm saying this is, these are the two examples which are going to apply now on many different doses in cancer also. Okay, the dose and the drug selection can be done based on the genomic architecture of that particular patient, of that particular individual. Now coming to the next question, what treatment line or when to be started? So we did an experiment of endometrium receptivity. To simplify, I'll tell, okay, after doing all the exercise of oocyte preparation, sperm preparation, and embryo formation, selecting the best embryo which looks morphologically and genetically suitable, transfer when? That's a big question in IVF. So we try to answer this question because we know the endometrium has a very small window from 20 to 24 days as span of 48 to 72 hours. Only this 48 to 72 hours is the window of implantation. But normally, it gets shifted, either prepondered or postpondered. Why? Because of the hormones, the medicines, the drugs which has been given to that female. This window of implantation heavily varies from individual to individual. So to subject this problem, we designed an experiment. We collected the endometrium tissue from the uterus of the the female subjected to infertility clinic, and we did the RNA extraction. We did a lot of exclusion, inclusion. We ensured that whatever we are looking at is the right tissue and the right space. And the RNA extraction then subjected to microarray and transcriptomic analysis of more than 13,000 genes. When we say 13,000 gene information, it becomes a lot of data. So we did a lot of QC matrices, ensured the data is appropriate, removed all the not so appropriate data or not what we call the noise, and then we started curating them into one single analysis. And what we saw, all the receptive females and the non-receptive females, the red ones and the yellow ones, they segregated very distinctly. They made a molecular signature of RNA expression. Based on this, when we deep dived, we could make much more small compartmentalization of these particular females, and how it helped the clinician. We now can report or predict what is the exact window of implantation to the precision of plus or minus 3 hours. I can, based on the report, tell to the clinician the receptivity of this particular female will be at 120th hour, 135th hour, 146th hour, or maybe less. So that precision helped the clinician to transfer at the right time, ensure the fertility or the pregnancy is positive, and that's how the IVF success rate increased from 70 to almost 92% now, with all the IVF centers that we work with. Now, coming to the fourth question, what approach should I take? What kind of approach should I take? This is one of the biggest challenges in clinical practice. Either I should go aggressive or I should go defensive. This is a big query which always haunts every clinician. And because he wants to see the patient should not run away, the patient should be safe enough, the new methods, whether they are appropriately validated, not. So all this question haunts a clinician. And that's where, again, the molecular approach comes into play. I'll give a very, very distinctive example for this. What we did, we know the fact that DNA, RNA, protein, and physiology, this is the information, this is a central dogma. But we also know what I am because of my DNA, what I'm being done, my age, my illness is due to the RNA and protein. But I also know there are influencers, there are external influences like bacteria. This microbiome, which is colonizing inwards or on the surface of my body, they influence a lot on my health. They influence a lot on my normal physiology. We started studying them. We took samples from different fluids, different part of the body, and see the diversity, how diverse it is, and how this diversity can be used to make some information. So what we did 130 female vaginal swabs we collected, and we started doing the microbiome diversity. And we started studying them. And once we did this through a 16S metagenomic next generation based sequencing, what we understood is every female from the age group of 12 to 45, that is starting of menses to ending of menses, is different from her age group, from her sexual habits, and from hygiene and everything. And then we started clinically correlating them. We found a group of bacteria and their diversity, abundance, I should say, that abundance is very much required for a fertile female. And anything other than that can be put as an infertile or not so good. Now, how does this information help? When we give this report to the clinician normally goes through that and says, okay, now diversity and the pathogenicity is there. Now, the clinician, based on the experience and discussion with the lab, they can take and call whether I should go for a rejuvenation by prebiotic and probiotics, or I should go for an antibiotic regime. So this offensive or defensive approach can be determined only by the information, the valuable information which we get from this kind of molecular approach. So in say, what I wanted to convey the message is, the information from different technologies, the genomics, the proteomics, the metabolomics, they all should be integrated, taken into account, new biomarkers identified, and that will give you the more broader aspect, more deeper insight of your health. So when we see through different lenses, we see different informations, and these different informations are too huge to imbibe, to understand, and to come to a conclusion. But when we overlay this information one above another, from the genomics, then the transcriptomics, then the proteomics, and the metabolomics, we get a better and a more wide picture. And when we do a cross-section of this information, we get exactly what we are looking for. And this information, when we do a cross-section, this information gives us what is happening in the body, how it is happening in the body, where it is happening in the body, and if we go much more higher and much more data with that, we can also predict when it is going to happen, when this particular problem is going to raise. Friends, I don't see less than a decade, you will all be having a genomic card of yourself, going to a clinician saying, this is my genome, this is the information of my genome, please subscribe me a drug and a dose according to my genome. You may seem this is a myth, a personalized medicine or customized medicine, but it is happening even today, particularly in cancer. In cancer, there are hundreds of different targeted therapy which are very specific to the mutation that you carry, or which are very specific to the driver mutation which has caused the cancer, or which is pathogenesis, which is the reason of the pathogenesis of cancer in your body. So when it has already started, we also need to adapt, we also need to accept, and we also need to move to this direction where this genomics and this different molecular approach are going to help us. So this is what we always tell in our lab. Knowledge is about right answer, but intelligence is about right question. So to the young mind here, I would like to always say, ask good questions. Ask more appropriate questions. Don't shy away from questions, however silly they may look like. But it has an answer, and if it has a good answer, your question was more appropriate than anyone else. This is our team who is working with me since last 15 years. We have been dedicated in new marker research, and thank you for your time, your attention, and your patience. Thank you.